Hello everyone. So in this tutorial we are going to learn about the cell cycle and proliferation. Why this cell cycle and proliferation is important in our body? Because it is very much essential, right? Cell proliferation is it is very fundamental in development and maintenance of the steady state tissue homeostasis. So that is a huge term. Tissue homeostasis means maintaining the constant number of cells in a tissue, and how it is doing, how that is maintained is actually by the replacement of the dead or damaged cell, by replacement of dead or damaged cells. So and what is cell cycle actually? Cell cycle means the sequence of events that results in cell division. So cell cycle is just a sequence of events which will result in the cell division actually. So now we can see that this cell cycle is regulated by activators and inhibitors. We have different amount, like different kind of these proteins, these enzymes which and these inhibitors which will control and will help in regulating the cycle. This is the cell cycle, and this cell cycle has the G1 phase. We have G1, G2, this is the G2, G1, S and M phase. So here G1 means the pre-synthetic growth phase. This is the pre-synthetic growth phase. That is why we have called it the G1. Then after this G0 is actually the quiescent phase. Quiescent cells, the cells which do not grow later on or for any kind of damage or checking the quiescent, the cell enters this state where the cycle gets the enough time, sufficient time to repair the damage that has occurred while replicating, so or while during the cell division, any kind of damage. So after this G0 or quiescent phase, we have let us see, this one G1 will progress to the synthetic phase, so which is called as the S phase, and here different synthesis will occur, DNA synthesis will occur. And then after we have this G2 phase, which means pre-mitotic growth phase. So this is the pre-mitotic growth phase, right? And M for mitosis. So this is the sequence we must remember, that G1, G2, these are the growth phase, and S and M, these are the synthetic and mitotic phase, and this will further progress in the cell division. So we will further see in different slide as well, in another slide also, and that would be the diagram from the Robbins pathology. So first of all, before knowing that, let me give you an overview on how these activators and inhibitors are acting, and what are these activators and inhibitors actually. So this regulation is done by the activators. So let us talk about the activators first. So this is now progression, the cyclins, these are the cyclins, and that is cyclin-dependent kinases. These are the enzymes of phosphorylation. So remember, wherever we hear the kinases, these are the phosphorylating enzyme. These are the phosphorylating enzyme which in order activate the other function in our cell or body. So cyclins, cyclin means these are the proteins which will drive the cell division, and these are usually of different type, but during the cell cycle we can preferentially see the cyclins of type A, B, D and E. And then we have the cyclin-dependent kinases. These enzymes are associated with cyclins and forms complexes at a different stage, or phases in the cell cycle. So we have cyclin-dependent kinases which are clearly visible during the cell cycle is cyclin-dependent kinases 1, 2, 4 and 6. We will call it the CDKs. Then after, what we have to see that if there are activators, then we also have to see there are inhibitors then. So before knowing the inhibitors, we will try to know how these cyclins and cyclin-dependent kinases works together. So see, this is the cyclin A, and this is cyclin B, cyclin D and cyclin E. I have made this connection, the complex in one diagram, but in reality this cyclin A will form complex different, like singly with the CDK1, cyclin dependent kinase 1, and another complex with the cyclin dependent kinase 2. So for just to make us easy to remember this, I have made it depicted it in a one diagram. So cyclin dependent kinase 1 and 2 forms different complexes with cyclin A, and that regulates the synthetic phase in the cycle. This is the synthetic phase. So these are the activators. This promotes and helps in the progression of cell division. So cyclin A forming complex with CDK1 and CDK2 differently, they will in a separate way they will help in activating the synthetic phase and regulating it. Then after we have cyclin B. Cyclin B will form the complex, like this will be complex with cyclin dependent kinase 1, and that will help in the regulation of this transition G2 to M transition. From this transition we will see the action of cyclin B and CDK1. So these are things to remember, such as remember A and B. So A and B are forming complexes with 1, 2, and this is the 1 again. So A, for additionally it will contain the cyclin dependent kinase number 2. So this is additionally, A for additional means number 2, cyclin dependent kinase. So let us move forward by knowing now we have the cyclin dependent, the cyclin D now form complex separately with cyclin dependent kinase 4 and cyclin dependent kinase 6, and that will help in this phase G1 to S transition. Now to progression regulates this cyclin D regulates the progression of the cell division from G1 to this S. At this phase the transition is controlled and helped by the cyclin D forming complex separately with 4 and cyclin D forming complex separately with 6. So this is the G1 to S transition. Similarly we have the cyclin E, which will also act in this forming complex. Cyclin E forming complex with the CDK2 will also be acting in this G1S transition phase. So it will help in progression and transition, like movement of G1, progression of the cell division from G1 to S phase. So this is all about the cyclin A, B, D and E, which will form different complexes with these cyclin dependent kinases for their proper activation of the and progression of the cell cycle that maintains the cell division and regulates the cell division in a proper way. Now we have the inhibitors. So inhibitors are usually known as cyclin dependent kinase inhibitors, which are of two group, usually two family. It consists of two families. So family 1 consists of the protein, this is P21, P27 and P57, which will act like broadly to inhibit all the CDK, almost all the CDK which are 1, 2, 4 and 6. So it will inhibit broadly all the cyclin dependent kinases in almost all the phases of this cell cycle. Now we have this P15, 16. Now another family, this is the second one family with P15, 16, 18 and 19, and they will specifically act and affect and inhibit the CDK 4 and 6. So where are the cyclin dependent kinase 4 and 6 forming complex and acting? So they are acting in this G1 to S transition. So this group of family of inhibitor, cyclin dependent kinase inhibitor, will inhibit the G1 to S transition. So this is about the cyclins, the inhibitors and cyclin dependent kinases. Now let us see in another picture where we can clearly demonstrate this. So this is the continuous, like we can see. Let's start from the cell division from the G1 phase. So growth in mass, centrosome duplication will occur, and here we have got the restriction point, and G1 to S is another checkpoint, while G2 to M is the another checkpoint in the cell cycle. So there will be the chromosome duplication and there will be DNA synthesis, and then it will progress through all this we have studied just a few minutes ago, and then the mitotic. Now completely, which are the cells which will not further divide, will stay as after one complete division. the permanent cell will be stay like that, neurons, cardiac myocytes. Similarly, if there is any kind of damage, or the cells which are now trying to stay in the quiescent or stable state, so that would, for example, we have got the hepatocytes. So from division, mitotic division to G0, now they will stay stable. But if there is any kind of damage, then it will again progress and be checked and repaired, and then it will move forward in this cell cycle. Now after this, we have to learn about and see the depiction of the inhibitors. So let us see how the inhibitors are acting. As I told you earlier, we have the two group of inhibitor and activators are cyclins and cyclin-dependent kinases. So see, this is cyclin D and cyclin D, forming complex with CDK4 and 6 and acting to the G1 to S transition. Similarly, we have cyclin A, forming complex with 2 and 1 and acting at the S phase. Similarly, we have the cyclin B and cyclin-dependent kinase 1, which is acting at the G2M transition, which I have already told you in the previous tutorial, that previous minutes, with a few minutes ago I told you. So this is another one. This is the cyclin-dependent, that is kinase 2 and E, forming complex and acting in the G1 to S transition. So this is all about the kinases and cyclins. Now the inhibitors. So we have studied that these are the P15, 16 and 18 and 19. These are the specific inhibitors of these complexes, cyclin D, 4 and 6 complexes. But these 21, 27 and 57 inhibitors will broadly inhibit almost all these stages and helps in progression, and that is important for the checking of the cell cycle or the faulty DNA replications, and that will be checking to prevent the mutation or any kind of cancerous cells. So this is all about the cyclin and cyclin dependent kinases and their inhibitors, and this is all about the cell cycle and proliferation. Thank you for listening me. Do comment below, and for any queries do comment below, and we will go further, continue the different topic and conceptual topics of pathology. So do subscribe my channel, and see you again in the next tutorial. Thank you and bye bye.